The next speaker would be Dr. Mohsen Mosayebi-Samani from IFADO in Germany. Mohsen, could you hear us?
Yes, and you can hear me as well?
Perfectly well. Go ahead.
Great. Thank you for attending to this talk and happy to have you here. Today I would like to talk about one of our recent study. A little brief introduction. As you might know, while there are a lot of promising results in NIBS application, the efficacy of the technique is still limited. One might explain this is the fact that our information about the optimal dosage and or stimulation protocol at the individual and or at the group level is still limited. Okay, one likely solution is individualized dosage adjustment according to the brain anatomical, brain state, or brain anatomical characteristic, for example, using electrical field modeling. But whether and to what extent the computational modeling explain the neurophysiological result of, for example, here in our study, tDCS, is still unknown. That was what we wanted to investigate. We had a data set of two subsequent experiments in which 29 participants were divided into two groups of anodal and cathodal tDCS, and in each group, participant received 0.5, 1, 1.5, 2 milliampere, and sham for 15 minutes. The first experiment was investigating neurophysiological effect of tDCS using TMS-MEP up to 2 hours after the end of stimulation in comparison with sham and or in comparison with the baseline. And then the second experiment was investigating the effect of tDCS using ASL fMRI. And then same story here, comparison with baseline up to or sham up to 2 hours after the end of stimulation. Participants were same in both studies. Next slide. Okay, for the effect of tDCS evaluating by TMS-MEP, we find that in case of anodal tDCS, almost all the intensities resulted a corticospinal excitability enhancement. But for cathodal tDCS, only 1 milliampere resulted in corticospinal excitability reduction. For the CBF, or cerebral blood flow, measured by ASL fMRI, we find a kind of linear enhancement of or reduction of CBF after tDCS by increasing the intensity. The question was that why? Next slide, please. Yes, for that, we tried to investigate if the computational modeling can explain this. For that, for each individual, we design a 3D model of head, and then we calculate the tDCS-induced electrical field, whether it was normal component, it was magnitude of electrical field at different region of interest. One of them was like a conventional, typical sphere under the target electrode, or the other was where the TMS induced the most effective effect under the coil. And then we also measured or calculate different anatomical measures like skull, CSF thickness, CSF volume, or electrode to cortex distance. Now we have a pool of measurement, physical measurement, and then we try to find out if there is an association between each of them and tDCS neurophysiological effect, TMS-MEP and or ASL fMRI. Can you go to the next slide? Okay. Our result in general showed that among all the anatomical measures, ECD, electrode to cortex distance, and CSF thickness could negatively and electrical field positively explained the MEP and or CBF alterations caused by tDCS. However, what we also found is that this result could be only valid when the tDCS induced significant neurophysiological effect, meaning that it might not be simply predict the tDCS response or neurophysiological response, at least in our case, in some dosage where we have kind of non-linear response. Next slide, please. Yeah. Okay, this was the result, but you should keep in mind that our result was only on motor cortex, and still the transferability of this result to the non-motor areas is under question, because our recent TMS EEG study, which is conducted here, but it's a preprint released. Previous slide, please. Still, yeah, showed that there is not a one-to-one transferability between motor to at least prefrontal cortex that we have tested. We have also a little bit evaluated whether the result that we obtain can be transferred to the elderly. At least the result is not published yet, but we didn't find a very nice transferability. The question would be that, okay, we did some correlation so far, a lot of other people did it as well, but can we do, as other previous presenter also said, should we do modeling first before applying before experiment, meaning that model-derived dosage adjustment can really improve the efficacy of the technique? For that, we need to for sure confirm or validate the computational modeling result, at least at the physical level, which is a big challenge and ongoing topic. And thank you for your yeah.
Thank you, thank you very much, Mohsen. Thank you.
Yes.
Thank you very much, Mohsen, for your presentation.